Thank you so much for bringing me to this wonderful place and to have this opportunity, Eddie. It's great to be with so many colleagues and friends. And I must say that with Eddie, in addition to being friends, we share something very important. As Eddie likes to say, him and I come from the same alma mater, which is this place, Facultad de Medicina, Universidad de la República, Uruguay, where Eddie was gold medal in the early 70s. And then I was also trained there. I'm still there in the Department of Biochemistry. So in any case, I will be talking about the topic of free radicals, oxidants, and antioxidants in physiology and disease and aging. Let's see, okay. And this is an area that crosses really throughout all the biomedical disciplines, because oxidants, free radicals, and molecular oxygen and nitric oxide are really involved in most disease conditions and even the aging process. So just to show a first slide from our group, just to show you that during inflammation and during the aging process, we oxidize our tissues, we nitrosate our tissues, and we nitrate our tissues, among many other oxidative modifications that undergo at the protein, DNA, sugar, and lipid levels. And this is a good example of how an inflammatory disease, this is a lymph node coming from a patient from our university hospital, and we tested for an antibody against nitrotyrosine, which is one of the post-translational oxidative modifications associated to inflammation. We can see some of the cells which are highly immunoreactive against nitrotyrosine. This is a footprint of disease progression and severity and sometimes outcome, but in addition to being a footprint, many times is also a mediator of disease. And the other thing I would like to say, that this being a stable footprint that you can analyze by immunochemistry and also by analytical method, the formation of nitrotyrosine is revealing a history of the chemistry in the tissue that involves free radical intermediates, including tyrosyl radicals, which is a protein-derived radical, and nitrogen dioxide, which is an oxidized derivative of NO. So once we see nitrotyrosine in a tissue, we know, as far as we know, that there was free radical chemistry behind that. And of the many organelles and extracellular sources, and even exogenous sources of free radicals, such as pollution, UV light, xenobiotic metabolism, mitochondria, and I know that Salvador will get deeper into this. Mitochondria are really a main source and target of reactive oxidants because over 95 to 99% of molecular oxygen consumed by mammalian cells usually goes into the terminal oxidase of the respiratory chain. And there is also always a small flux of oxygen that instead of going directly to water, goes to superoxide radicals and hydrogen peroxide. And those levels that usually different measurements, including those of our group, range in the order of 0.1 to 0.2% of total oxygen going into mitochondria evolves into free radicals. These values are significantly lower than values provided some years ago. And that percentage can really increase. And mitochondria not only generate free radicals, mitochondrial components are targets of free radicals, and this is part of some of the actions opening the transition port to signal apoptosis under some conditions. But also mitochondria are emissors of hydrogen peroxide for signaling purposes, and this is a relatively new area. To visualize mitochondria as a hydrogen peroxide emissor to do redox signaling in the cytoplasm. So, different sites of mitochondrial superoxide radical and hydrogen peroxide formation, including complex 1 and complex 3 of the mitochondrial electron transport chain. This is a continuous process throughout the life that can be enhanced several-fold under many pathophysiologically relevant conditions. And of course, mitochondria are empowered with very strong antioxidant mechanisms, which are critical for cell survival and proliferation and inhibition of malignancies, such as the manganese superoxide dismutases, which are essential enzymes. There is no such a thing as a double knockout in manganese SOD. This is an essential enzyme. And also in the intermembrane space, there are the copper zinc SODs. And these enzymes get rid of superoxide radicals, transforming them into hydrogen peroxide. But now hydrogen peroxide, again, can be catabolized in mitochondria by peroxyredoxins and glutathione peroxidases, and in the peroxisomes also by catalases. So there are strong antioxidant systems that keep the steady-state concentrations of radicals and oxidants relatively low under physiologically relevant conditions. In some cases, radicals are utilized to kill bacteria invading pathogens and even tumor cells, and some of this is done by the oxidative burst promoted by phagocytosis and the activation of the NOx2 enzymes, the NADPH oxidases, and basically a pathogen engulfed inside a phagosome, and we have done extensive work for Trypanosoma cruzi and other bacteria. There is a lot of cytotoxicity inside the phagosome because of oxidative reactions. And at the end of the talk, I will show that the pathogens defend themselves by upregulating antioxidant defenses, and the pathogen antioxidant defenses constitute a virulence factor. More recently, and I know Salvador will also touch on some of these, other cell types, including vascular endothelium, can also generate superoxide radicals at the plasma membrane at much lower levels to regulate reactions such as nitric oxide bioavailability. And this can also have pathophysiological consequences in the blood vessels in conditions such as atherosclerosis and hypertension, where nitric oxide bioavailability is compromised by excess formation of superoxide radicals generated upon stimulation by angiotensin II or oxidized lipoproteins in excess in the circulation. So this is some of the ideas. There is a continuous consumption of oxygen to water by the cytochrome oxidase. This is a tetra-electron reduction process without release of any partially reduced oxidized intermediate. So the cytochrome oxidase doesn't release any oxygen until it's completely reduced to water, but there is a certain amount of electrons that leak out to molecular oxygen to form superoxide radicals, hydrogen peroxide, that then are catabolized by different antioxidant systems, and then in the presence of transition metal centers and other reactions, such as the decomposition of peroxynitrous acid, that I will mention in a minute, generate this powerful oxidant hydroxyl radical that there are no enzymatic mechanism for its removal, but it has to be eliminated by low molecular weight scavengers. Some of what it can be done, it was nicely highlighted by, which was my postdoctoral mentor, Bruce Freeman. In the 80s, proposed a mechanism of free radical damage in membranes and lipoproteins. This was a postulation. Now we have been able, we and many other groups have been able to measure many of these oxidation products that can be formed, and secondary products such as aldehydes. Aldehydes are being released secondary to lipid peroxidation, and aldehydes are electrophiles that can do electrophilic signaling and electrophilic toxicity. So it's not only an issue of oxidants, but then secondary intermediates such as electrophiles are generated throughout these oxidative processes. So just to go into the definition for those that are not so familiar with chemical aspects of the field, a free radical is any molecule that contains one or more ampere electrons in its upper orbital, including superoxide radicals, and in red you can see this ampere electron, and even nitric oxide, which is a central master regulator in cell and tissue physiology. It is a free radical, but while superoxide radicals are highly reactive and a very short life, in the order of nanoseconds to microseconds, nitric oxide is a pretty stable radical. In the radical world, it's a stable radical, and it lasts In tissues, it can last for about 1 to 10 seconds, as Salvador and many other groups have shown in vessels. For us, a radical that lives 10 seconds is an extremely long-lived free radical. Usually, the species we deal with, and we have to measure, live in the order of microseconds to milliseconds. That's about the time scale we usually work on in the laboratory, for which we need to develop fast kinetic techniques. Nitric oxide as a central regulator of different physiological actions, including vasodilation, neurotransmitter, it has also a dark side, and is the cytotoxicity related to nitric oxide. And much of our work over the last 2 decades has concentrated to understand how nitric oxide can shift from a physiological regulator to a pathogenic mediator. And much of this shift has to do with the fact that under normal conditions, NO goes into the upper pathway, which is reversible reactions with heme proteins, such as 1-L-8 cyclase and cytochrome oxidase, and these are reversible formations from nitrosyl iron complexes, leading to stimulation of 1-L-8 cyclase or transient inhibition of cytochrome oxidase, and this is physiology. But upon a shift in the redox status of the cell, including enhanced oxidation processes, NO shifts the chemistry into the lower part, and much of that is due to the reaction of nitric oxide with superoxide. This is a radical-radical combination reaction, which is diffusion controlled, meaning it goes as fast as a reaction can go. It outcompetes the superoxide dismutases for the elimination of superoxide. It regulates the biological half-life of NO and leads to the formation of a potent peroxide, potent and rather unusual peroxide, peroxynitrite anion that has a pKa around 6.8, meaning that under physiological conditions, both peroxynitrite anion and peroxynitrous acid coexist. The chemistries of these two species is rather different, but they promote nitration, nitrosation to some extent, and to a large extent, oxidation reactions, and I have already talked about the formation of nitrotyrosine. So the concept of oxidative stress was put forward by Helmut Sis in '85, updated in 2007, reflecting the development of the field in terms that redox mediators not only do damage, but they can do also signaling at low levels. So oxidative stress deals now with the oxidative damage, but also with the disruption of oxidative signaling. And around the same time, with Gonzalo Pelufo and taking into account many contributions from many groups, we sort of expanded the idea to nitrooxidative stress, where nitrating intermediates and nitric oxide-derived oxidants participate in this chemistry. And a very nice review by Helmut Sis a few years ago pointed in the metabolic role of H2O2 generation from redox signaling to oxidative stress. So now we understand much better the different phases in which these oxidants participate, and we do need some flux of oxidants to have a healthy life at the cell and tissue level. So the redox homeostasis has a low flux of oxygen, is important for signaling, and the redox-sensitive transcription factors and signal transduction and gene expression. And then if you go through over some threshold, then oxidative stress and oxidative damage start to predominate. Some of the failure of some of the clinical trials utilizing mega concentrations of, mega levels of antioxidants for patients is more likely due to the fact that it's interfering with the signaling actions, in addition to blocking the oxidative damage. How do oxidants transduce signal is a big kinetic problem. I will not get into any detail into this, but just to highlight some ideas that we put forward in the early 2010s with Gerardo Ferrer-Sueta, and just to show you that redox cascades can lead to the activation of, for example, NRF2 system, but if in excess, also NF-kappa B and pro-inflammatory responses are activated. So this is part of the issue, and there are some kinetic bottlenecks, because once you generate an oxidant, there has to be some sensors that will react at significantly faster rates so that the fate is directly to some specific route. Otherwise, everything can react with everything in this world of these free radicals and oxidants. So we are starting to understand that there is a hierarchy on reactivities, and some of this has to do with extraordinary reactivity of the thiol-containing enzymes, the peroxyredoxins. These are fast-reacting thiol-containing enzymes that are around the first targets for hydrogen peroxide, peroxynitrite, and lipid hydroperoxides. And then the peroxyredoxins can, on one hand, eliminate and catabolize oxidants, but on the other hand, they can constitute redox relays, as Tobias Dick in Heidelberg has put forward recently. So there is a peroxide reacting with a fast-reacting thiol, and we and other groups have determined many rate constants of these reactions, which are in the order of 10 to the 7 per molar per second, a rather fast reaction for peroxides and thiols. And then once these relays react with thiols, you generate sulfenic acids. And then the sulfenic acids can be resolved internally by the catalytic cycle of the peroxyredoxin, or alternatively interact with redox partners to generate mixed disulfides that then translate the redox reactions to a secondary redox mediator and then to a third redox mediator until a redox-sensitive transcription factor gets the signal and then migrates to the nuclei to trigger genetic responses. So now we are starting to understand the kinetic basis of redox signaling and redox decompositions. And I will get back to the peroxyredoxins because they do this in mammalian cells, and they do this in the cytosol and in the mitochondria, but also the pathogens overexpress peroxyredoxins to cope with the toxic effects of oxidants generated by the immune response. So a decade ago already, we put together a review article where we put some of these ideas together in terms of the shift of the physiological chemistry of NO towards pathology, and many different groups have contributed and have found that this mediator participates in inflammation and sepsis, neurodegeneration, cardiovascular diseases, and immune responses, and we'll touch a little bit of this in the next few minutes. A central mediator of all of this chemistry is mitochondria. This was a very nice cartoon generated by Valeria Valles in our laboratory when she was finishing her PhD thesis. And basically, this is showing NO, this is the white cloud, diffusing into mitochondria. NO is a hydrophobic, neutral, and a small molecule that can easily diffuse across cell membranes, and mitochondria is a continuous generator of superoxide radicals. So inside the matrix and in the intermembrane space, you get the right mix of NO and superoxide, and leads to the formation of the yellow product, which is peroxynitrite. And there is a lot of action of peroxynitrite inside mitochondria. And this has to do with alterations in mitochondrial bioenergetics and also the opening of the permeability transition pore and signaling of apoptosis. So some of the chemistries we have been studying and applying are compounds that can do things inside mitochondria with antioxidant capacities, and these are catalytic eliminators of superoxide radicals, and more than that, peroxynitrite. And these, for example, are the manganese porphyrins, and we have done this work in collaboration with Duke University, Ines Batinik-Haberle. These are the organic chemists that synthesize all this variety of manganese porphyrins, which are tailored, changing the lateral chain to fine-tune the redox potential of the manganese and the hydrophobicity. And this can be utilized in vitro and in vivo, and some of these compounds are now in clinical trials for a variety of diseases. And we have tested these compounds in a variety of preclinical models. The manganese porphyrins are administered in the manganese 3+ form, but the bioactive form is the manganese 2+ for this manganese porphyrin to become an effective catalytic antioxidant. So one of the questions is, who is doing the reduction of the manganese 3+ to the manganese 2+ in vivo? We are still working on this, but just to show some evidence, the electron transport chain is doing this. So the manganese porphyrins can reach mitochondria, and at the expense of electrons flowing through the electron transport chain, become reduced and bioactivated there. So these are excellent protectors of mitochondria, utilizing electrons that otherwise come from NADH or from succinate, because both complex 1 and 2 can reduce the manganese 3 to the manganese 2. And now once it's the manganese 2, you can see that the manganese 2 can then react with different oxidants, including peroxynitrite, and then eliminate it to nitrite, which is rather inert. And then it goes to the manganese 4. And sacrificial antioxidants, such as glutathione, uric acid, ascorbate, reduce the manganese 4 back to the manganese 3, and then in the manganese 3, the electron transport chain and other flavoid proteins put them back into the manganese 2, and then with micromolar levels of the manganese porphyrins, you can really eliminate huge levels of oxidants generated into mitochondria. So different models have shown effectivity of these compounds, and I will just show one of them, which are the motor neurodegeneration. This work has been done in collaboration with a very gifted neurobiologist and friend in Uruguay, Luis Barbeito, from the Pasteur Institute, Montevideo. This has been a very important collaboration through the years. And then we know that motor neurons, upon growth factor deprivation, die via apoptosis, and this pathway involves the formation of peroxynitrite and oxidative stress, and all the ways we have blocked this is work that has gone through the last over 10 years. And then we put together the model, the animal model of ALS, which is a transgenic animals expressing the gene of a mutated copper zinc SOD form that is present in familial forms of ALS. And then the rat and the mice develop an ALS-like disease after 90 days. So in this model, we apply the technique of immunospin trapping, and this was the first application in vivo of the method by which animals administered with EMPO, the protein radicals were trapped and revealed immunochemically. And basically, what's shown that in the spinal cord of these animals, the motor neurons of the sick animals, in the left you see healthy motor neurons in the anterior horn of the spinal cord, these are fine, and in the central panel you see dying motor neurons which are immunopositive for the anti-DMPO protein adduct, meaning that there was free radical chemistry there. These neurons are also nitrated, and when we utilize the manganoporphyrins and isolate mitochondria, these are mitochondria from brain and mitochondria from the spinal cord, the level of oxidation is significantly less. If we move one step forward and we start treating animals after the disease started with a mitochondrial targeted antioxidants, such as the MitoQ, work in collaboration with Mike Murphy that sent us the compound, which concentrates into mitochondria about 500-fold via this triphenylphosphonium moiety that is incorporated utilizing the electrochemical gradient. And then these animals, by oral administration of MitoQ, live longer, and also in the grip strength test, and they have more strength in their upper limbs. So it works. And this was just one single, it was just one dose. This possibly can be improved with time if we utilize other levels of mitochondrial targeted antioxidants. So just to finish in the last minute, to show you that these nitration reactions are important in aging, and this is a wonderful piece of work that I just brought because I think it's great, by Professor Volker Ulrich and collaborators in Germany. They see that vessels throughout life decrease their responses to NO, and some of this is to the enhanced formation of superoxide by the vessels, but not only NO is disappearing, but peroxynitrite is being formed, and this is the immunogold labeling of nitrotyrosine in the vascular endothelium. I don't know if you can see well, but you can see the paper. Many of the organelles are nitrated, the dots are gold particles, and especially mitochondria is nitrated throughout the aging process. And this is normal aging process. This is not pathology. Just to end, to show you two applications. I will not get into any detail, but these are work we just published in this last year. in antioxidant enzymes of different pathogens, including Trypanosoma cruzi, and then the lower Pseudomonas aeruginosa, and we find that the antioxidant systems in the pathogens constitute virulence factors. And this is a review we wrote a few years ago, peroxynitrite, a stealthy oxidant. So this is the Bay of Montevideo, and I would like to thank all the people that have mentioned and the funding agencies. Thank you very much.